Alright, everybody. Sorry about that. I have figured out that there's about a 20-25 second delay in between what I'm saying and what you guys are picking up. So I do apologize for that. YouTube will not let me pick the ultra low latency. I don't know why. Normally it does. Now it's not. So we're just gonna have to deal with it, and things will just be a little laggy, I guess. A little laggy. All right. So today's just kind of a Q&A session. See, there's 4 of you guys on right now. I mean, we can go over anything from how to do like standard deviation and standard error. We can go over another FRQ. We can go over some of the information I've heard through the grapevine about AP tests in general. Nothing too specific. Don't worry, College Board. I haven't received any specifics about what has been on the test, just kind of, what some people have noticed trend-wise with the test. Yeah, so down below is the comments. That's where I'm going to be looking for questions you guys may have. If I say something like, oh, I want to know more about that, type it into the chat, and I'll pause what I'm doing to address any question you have. I know somebody texted me a question like 2 minutes ago. I'm like, just ask it on the stream. I mean, I don't want to do it right now. I think that's it. Fun fact, I'm wearing a headband, Gokaku. I purchased it in Japan. It's for Success for Testing. And so I thought it'd be a really fun thing to wear during AP testing time. I was going to wear it like morning of, but, we didn't. We're not doing it like that this year. We didn't get to hold a pancake breakfast like I've done in the past, which kind of breaks my heart. So maybe what we'll have to do is I'll have to invite all the AP back next year for chocolate pancakes. All right. So, again, just waiting for any suggestions you guys have, what you guys want to go over. Are there any formulas you want to go over? Again, standard deviation, standard error. Information. Tests. There's a really bad delay. Like, I'm just now seeing me pick up the water bottle. It was a while ago. This is getting awkward. Ha 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 ha. There you are. Wondering where you're going to be, Max. All right, so Max has had the question. How is ATP regenerated from ADP? So remember the structure of ATP versus ADP. And actually, let me pull you up a picture right now. ADP versus ATP. ADP stands for adenosine diphosphate, and ATP is adenosine triphosphate. So, I like this image a lot. Open image and new tab. I had to pick a tiny one, didn't I? Click this one. Any bigger? A little bit bigger. All right. So adenosine diphosphate is 2 phosphate groups, and adenosine triphosphate is 3 phosphate groups. So if we're looking at this ADP molecule, I really need a bigger picture. Hold on. Actually, we'll just use this one. Nope, that's also a tiny picture. Thank you, Google Images, for tools. Yes, size large. Look, big one. Yes, it is. Open image, new tab. It's giving me some trouble. That's not a large image. Oh, this is fun. Totally not awkward at all. Open image, new tab. There we go. Move. There we go. All right. so let me turn off document camera, webcam, transition. There we go. All right, so we have our ATP, our ADP. So ADP is adenosine triphosphate again. ATP is adenosine tri, so 3-phosphate. ADP is adenosine diphosphate. So in order for that last phosphate group to attach on, we need to do dehydration. Dehydration synthesis to attach that last, or not dehydration. Anyways, we need to be able to attach that last phosphate. Now this happens through a molecule called ATP synthase. ATP synthase is located in the walls of the mitochondria. And what happens is, let me pull up one of those fancy diagrams. So ATP. Oh, they got a really cool picture of it, but it's not the picture I want. Images. Ah, this is a good one. Nope, it's a terrible one. It is not. How about this one? This one? These are all really low resolution pictures. Hold on, guys. It takes me a second. Why can't the internet just be nice to me? Goodness gracious. So sorry. I didn't think I'd need a picture so early. It's a terrible internet. Let's do this one. Nope. Yes. Ah ha ha ha. Down. Okay. So, what's happening, and if you look at the bottom right hand corner, this is actually where ATP or ADP turns into ATP. So here we go. So if you remember during cellular respiration, we make that NADH molecule. It's normally NAD plus, but we add that electron onto it to create NADH because now it has a proton and it also has an electron on it, so it evens out in charge. But that electron, all it's doing is just hitching a ride on NADH until it gets to the electron transport chain, which is this step right here. And so we have our different, I can't zoom in that far. We have our different systems in place for that. And what's going to happen is as it drops off those hydrogen ions, they're going to be pumped across the membrane. So there's an area of high concentration, those hydrogen ions, an area of low concentration on the outside. What's going to happen is those hydrogen ions are going to want to pop back out, specifically because this oxygen right here, this oxygen is going to be so electronegative, it's going to be pulling these hydrogens through the electron transport chain for the sole purpose of turning into H2O. Okay? Then the hydrogen ions that diffuse back through, this molecule, let me see if I can find a cool picture of what ATP synthase actually looks like. Aha, here we go. Okay, so open image, a new tab. All right, so this is what ATP synthase actually looks like. All right, it is a bit of a revolving door up here. So what's going to happen is as those hydrogen ions pass through ATP synthase, as they pass through, they're going to be turning this wheel right here. Now, when you turn that wheel, it's actually going to produce energy, because you know energy, which is ultimately what's going to be used. That energy is going to be converted into chemical energy. It's going to be the bond between the last two phosphates of ADP as it turns into ATP, which is kind of cool. Isn't that awesome? I love it. I love it so much. Very underrated as a molecule. Did that answer your question, Max? Oh, we gotta wait. And so the question for Ghassan is, what do you mean by gene regulation, because there's a bunch of different types of gene regulation. So it really kind of depends on what you mean by gene regulation. Let me grab a book. Oh no! All right, and I have, oof, I won't be able to tell you what chapters are on the book topics because I don't actually have one of those. I don't have one of those books at my house. I had one up until probably about a week ago, and then I took it back because I have the new version here. I'm like, oh, cool, I got the new version. And so I can give you the books for or the chapters for the new version, but not the old version. That being said, let me check my Google Drive. So ATP is ultimately created by the energy that is formed during that twisting motion. So it's like a kinetic energy is being transferred into a chemical energy. Now, some of the efficiency is lost as heat, which is great because we stay warm all the time. Doesn't matter if you're in Russia or wherever. But that kinetic energy is being transferred into chemical energy, which is specifically that bond between the last two phosphates. Because remember, energy can be transformed, it can be changed, but it can never be destroyed. Laws of thermodynamics. Okay, let me see. Chapter-wise. Alright, so, Gassan, for your guys' books, chemistry is chapters 2, 3, and 5. Cells is 6, 7, 11, and 12. Cellular energy is 8, 9, and 10. Heredity is 13, 14, 15. And then DNA is 16, 17, 18, 19, and 20. We don't have to worry about 21 this time. So just 16 through 20 for that. Just looked up some old files. So cellular respiration, all you're doing is just going from ADP to ATP, ADP to ATP. It doesn't make it. It doesn't make the whole thing. It just recharges it, kind of like the battery on your phone. You don't get a new battery every time you plug it in. You're just recharging it. Also, Gassan, what did you mean by gene regulation? Because there's depending on how far you want to go with gene regulation, there's a lot. Here. Repressible versus inducible again. All right, so the difference between repressible and inducible. Repressible genes are one that you can add something to it to stop it from being formed. And inducible is you add and by something I mean like a ligand of some sort, a signaling molecule, something that will bind to like the operator region of DNA. So maybe it will bind to like depending on if we're looking at like lactose or whatever, it could bind to a molecule to then allow it to be produced. So this is actually the case of lactose. Let me see if I can get you guys a picture, because they say a picture is worth 1,000 words. Images. Aha, I like this one. Okay, open image in new tab. we go. Picture's worth 1,000 words. Okay. Transition. There we go. So repressible is when you can stop it, the gene from being expressed. Inducible is when you start it from to be expressed. So for instance, we have a lac repressor. So this DNA is repressible until, although it says lactose binding to the lac repressor is actually a, oh, what do you call them? An isomer of lactose that actually will cause the lac repressor to then be removed, which is kind of interesting, actually. Which is kind of interesting. Oops, I am losing track of my tabs. Here we go. Oops, wrong tab. Alright, yeah. Does it need further explanation than that kind of whirlwind of an answer I gave? Gotta wait. Sorry. Again, there's like a 20-something second delay between what you guys are hearing from me and what you guys and what I'm actually doing. So this part up here is going to be repressible, and this one down here is inducible. Lactase is actually both, which is kind of a weird thing. Tyrosine kinases. All right, T1 receptor tyrosine kinases. Pull up an image. So in this I guess this does. When lactose is added to the inducible operon, then RNA polymerase. Yeah, so it's actually when a isomer of lactose is introduced. It's not actually lactose itself. This is technically incorrect right here. When that is what will bind to the repressor to then cause the repressor to change shape and then pop off, which will allow the RNA polymerase to then zip through on that DNA. But until that isomer is present, it won't happen. And the reason for this is you don't want to be creating lactase when you don't need lactase. Enzymes are kind of expensive, so you don't want to be, buying them or making them if you don't have to. It's kind of like if you're buying like really expensive oil but you don't have a car. What's the point of buying, fancy motor oil if you don't have a car? So what's the point of creating lactase if you don't have any lactose in the system to digest? Yeah, so the repressor itself is the repressible portion, and the inducible is that isomer of lactase, because that will now induce, it will start up the lactase production. Yeah, so before lactose is even present, the repressor is attached, and again, it's just stopping it from being transcribed because you don't need it yet. So the lac repressor is preventing everything from moving forward until lactose is present. Just waiting for a response for that. Excuse me. Tastes like El Pollo Loco. All right. So heading back to Max's question, because Max, tyrosine kinases. Um, I would think it'd be pretty open image in new tab. This one. So here's actually the picture from our book. I do think it would be maybe not rare, but yeah, it was rare that they asked such a specific question. Although, if I am correct, give me 1 second to pull out a fancy book. And that. Oh, ow. We're good. Okay. Okay, so if we're looking at specifically what AP has delivered to us as far as what needs to be covered in the AP course, it talks about G protein coupled receptors are an example of a protein receptor in eukaryotes. And it talks about the 3 steps to cell signaling. So, you know, I have the ligand that binds, second messengers, and then the final induced response of the cell. Signal transduction pathways, mutations in those pathways, what can happen. Yeah, so it doesn't specifically talk about receptor tyrosine kinases, so that is out of the scope of this class. However, that being said, they're a fantastic example of a receptor on a cell anyway. So if we're looking at it as an example, so we have, what's going to happen is we have our signaling molecule over here, all right, and these are the signaling binding sites. Now remember, the binding sites are very, very specific to the ligand. You want this, you want to be specific to the signaling molecule, otherwise anything and everything is going to trigger this receptor to go off. And so you wouldn't want any little pressure, which is a different signaling molecule than pain, you wouldn't want any little pressure to cause pain up your arm instead of, oh, I'm just feeling that I have pressure on my hand. So the actual signal is very, very important, and also the receptors. They are very, very specific to what they do. All right. So here we have a receptor tyrosine kinase. And so what's going to happen is when you have 2 of those signaling molecules attached, it's going to cause the enzyme to dimerize. Now, dimerize means that they come together. You can actually have dimers in your DNA, which actually is a very common side effect of UV radiation. Is dimers, they're called thymine dimers, where thymine next to a thymine will bind together. So instead of binding across from them on that ladder, they'll bind sideways to each other, which is bad, causes issue with DNA, and as you know, too many issues with the DNA can cause cancer. Anyways, so it will dimerize, right? Here's the dimer being formed. And then when the dimer is formed, it causes the tyrosine kinases domains to become activated. All right, oh look, they're activated. Check that out. Now you do need energy in order for this to happen. And so when ATP turns into ADP, it leaves those phosphate groups attached. So they call it phosphorylated. Anytime you have the word phosphorylated, it basically means a phosphate group was added via ATP. Now, when you add on any chemical, any element, anything, a phosphate group, to a enzyme, to a protein, to whatever, what it physically does on the molecular level is it changes the shape of the molecule itself, which will cause it then to do a job of some kind. So for instance, once the tyrosine gets, the tyrosine kinases are phosphorylated, they have a phosphate, they change shape just enough to activate relay proteins. Now, those relay proteins can't normally attach to the tyrosine kinases unless they have that extra phosphate group, which changes the shape of them and allows that lock and key fit. All right. And then once those proteins are activated, then it just starts that cascade where this will activate this, which will activate this, which will activate this, which will activate this, which will then finally cause a particular, I don't know, protein to be transcribed from the DNA and then translated in the cytoplasm. Did I answer your question, Max? There's a big delay. I'm sorry I can't do anything about it. Well, let me click ultra low latency. I guess I gotta give up my dreams of being a streamer. I'll never be mumbo jumbo. Yep, thank you. Alright, cool. Alright, any other questions? So one of the things I suggest you guys doing sometime within the next, what do we have? Friday, Saturday, Sunday. 3 days, okay? There's a couple of things I want you guys to do to prepare for the test. One, make sure you have your equation sheet ready to go. I suggest printing it, but I am a little on the kinesthetic side, so I want to be able to touch the papers I am holding, which is why sometimes I don't particularly enjoy like digital assignments, because I want to be able to touch them, to grab them, to write on them. That's just because that's part of my learning style. But I mean, if you're totally not a kinesthetic person at all, just make sure you at least have what are they called? All the documents downloaded, ready to go. That way, it's at your fingertips right away. Electron transport chain. All right. All right. While I look this up, can't type and talk at the same time. Anyways, I'm going to continue with what I was saying. So make sure you have your equation sheet ready to go. I don't know if they're going to have it up or not. You guys are welcome to my anything I've posted on Google Classroom to have it ready to go for you guys, including that little thing we went over last time in the last stream of like basically just the class notes. Have everything out ready to go. Nothing will be more frustrating to you than if you only have that time and you can't find that thing that you need. I don't know about you guys, sometimes I'm a little disorganized and I can't find that one piece of paper. I can imagine in my head and I'm like, oh yeah, I totally had it, but where is it? Have everything out ready to go, okay? So make sure that's all out. Make sure you have your equation sheet out. I'll get to the questions in a second. Make sure you have anything and everything you need. A bottle of water is always nice. Don't drink too much. Please go to the bathroom before the test so you're not getting that anxious, I gotta go pee, I gotta go pee. Because that was always something that bothered me for testing in college. I always made sure I used the bathroom. I always made sure I was fed. Not too much, because you don't want to be sleepy during the test. Because what's going to happen is the blood flow is going to go to your digestive system to help digest that food. Just make sure, like, you're just topped off. You're not hungry. You're not full. You're just right. And that's something I would always make sure I was doing in college right before an exam. Make sure I had everything. I was full. I had a good night's sleep. Try to get to bed a little early that night. It's going to take you a while to get to sleep. Yeah, no, if you can't find your notes, you can use the internet. Please, please, please avoid, what's his name, Dinosaur Sauce. Whoever it is on the internet that's trying to get everybody to cheat on the AP exam. Don't listen to him. Just Google what you need. and be done with it. Do not copy and paste. They will know. They have plagiarism checkers, so you want to be all in your own words. Anyways, get a good night's sleep, or try to at least. Um, make sure you're in a comfortable environment. Like, make sure you have maybe a nice, soft, cushy chair. Not too comfortable that you fall asleep in, though. Comfy chair. You're wearing comfortable clothes. I suggest pajamas. Although those might make you sleepy if they're too soft. Yeah. So, I mean, I never went to Cal Poly in pajamas, but I was made sure I was wearing comfortable clothes. I have a couple shirts that are really soft, and so I like to wear those for test days. Because they help me out, because now I'm not thinking like, oh, that tag is itching me while I'm trying to take a test. None of that's bothering me. All right, so just a couple pieces for you guys to think about. All right, so some of the things we're gonna go through the electron transport chain first. And so I said there's a few things that might be on the AP test. Yeah. So it seems like, and this is kind of what I thought they were gonna do from the start. Just because you have to have a way in order to separate all the different scores of all the different students, and how do you quantify a 1, a 2, a 3, a 4, a 5? Because normally during the regular AP exam, I mean, you have 60 multiple choice, 2 long, 4 short for your response. So the amount of points to be able to divvy up into what points will earn you a 1, what points will earn you a 2, a 3, a 4, a 5. So in order to counteract the fact that they don't have a ton of questions to throw at you this year, what they're gonna do is they're gonna throw a lot of questions at you. All right, they might throw more questions than you physically have time to access. All right, especially if we're looking at the like the example they posted. Oh, extracellular matrix, got it. Oh, I was thinking ETC, not the ECM. Anyways, I'll look that up right now. I'll pull up some images. Anyways, what was I talking about? Yeah. So I think they're gonna give you guys more questions than you can shake a stick at. So what I suggest doing is you read the questions first, pick the ones you know for sure, or the ones that you think are gonna be the easiest. Answer those first. Then answer the ones that you think like, oh yeah, I probably know this one. And then save the ones you're like, I'm gonna have to pull something out my butt for this. Save those for last, all right? Because you don't want to be wasting time trying to do them in order when you don't really know kind of how to do question C. You know a little bit, and maybe it'll come to you, but it's just wasting time at that point. But yeah. So answer the questions you absolutely know first, all right? 25 minutes is not a lot of time. Not a lot of time. And when it hits 5 minutes, it's going to turn red. Submit your answers. Don't think like, "Oh, I can work for 2 more minutes and then have 3 minutes to submit." No, please. One of the things I've been reading online is some people have been waiting a little too long to submit, and by the time they actually get those files uploaded, it's too late. All right? So definitely when it hits that 5 minutes, just stop, turn it in. All right. One of the other things I've seen happening with people is formatting issues if they're using their phone. All right. So go into your phone settings and check to make sure it's being saved in one of the appropriate formats. I've actually heard that some iPhone users were having troubles because it was saving in a weird like HLM something format rather than like a PDF or a JPEG or whatever. So make sure that your phone, if you're using your phone or whatever device you're using, is making like a PDF, straight PDF. Like, can't go wrong with the PDF. That way, when you submit it, is in a file that is readable to College Board. Because if they can't read it, and it will physically stop you from submitting. And so I've heard there's been some issues of it not submitting because it's the wrong format and the system wouldn't allow it. Yeah, so make sure it's in the right format. If you are doing like a Word document and you want to save it as a PDF, because let's say you had to put a diagram on there or whatever, you're going to go to File, Save As, or Save, and then when it allows you to type in the name of the file, right below that, it'll actually allow you to change the format as well. And so sometimes I do that for uploading for you guys. So yeah, if you guys need help with that, let me know. Although it's pretty easy. Okay, let's see here. Yes, you can use the internet, and we're looking at extracellular matrix right now. All right, as far as College Board goes, excuse me, I got the hiccups. Size, membrane permeability. Make sure you know for the plasma membrane, we did do this on Tuesday with the diagram that you know there's glycoproteins, which is just glucose attached to proteins. That we have glycolipids, which is sugars attached to fats or lipids for the cell membrane. Membrane transport. Enzymes, cellular respiration. Interesting. All right. So I'll just talk then, because I can't seem to find what they. Golgi apparatus, rough ER, smooth ER, permeability, cell membranes, endocytosis, exocytosis. Know how to calculate water potential. Alright, it doesn't say. So instead, I'm just going to talk about the extracellular matrix. Okay. Teoglycan, open image tab. Alright. So this is a picture of the extracellular matrix. Now, the way I like to imagine the extracellular matrix is it's kind of like a suit of armor that you're wearing, like if you were a cell, that is attached directly to your skeleton. So you know you have like let's say a helmet, and there would be pegs that would go in and attach directly to your skull. That way it provides support and rigidity. And that's exactly what's happening on your extracellular matrix. There's collagen fibers in there, which are fantastic at keeping that spring in your skin. And also, when those collagen fibers start to break down, it's when you get wrinkles, which is why they inject collagen into people with wrinkles to try to get rid of them. That way, those fibers continue to be elastic in the skin and make sure it's nice and taut, sticking together. Proteoglycan, elastin, fibronectin, these are basically all just fibers that help to connect the cells to other cells, provide rigidity for the cell. It's kind of like an exoskeleton for the cell, although it does actually attach to the cytoskeleton on the inside too. But again, it's just providing support for the cell. I don't believe you'll need to know all the super like detailed parts, like integrins are kind of important. Think like integrin, like integrated. They are proteins that are integrated into the cell membrane that allow for support structure of the extracellular matrix to the cytoskeleton, not cytoskin, cytoskeleton filaments on the inside. All right, anything else? Yeah, so again, they're going to give you more questions than you might be able to answer in a 25-minute period, and that's okay. Just answer what you can. Remember, it's a numbers game. Generally, it's about 50% of the questions correct gives you a 3. So, I mean, I guess Thanos had it right in that idea. It's just 50%. That's all you need. Although he totally did disrupt the food supply chain when he wiped out half the world. But, you know, he didn't think that far ahead. Ooh, what does cholesterol do in the membrane? Cholesterol will actually, let's see if I can find a picture of it. The answer is no. Cholesterol allows the membrane to remain fluid at lower temperatures, because like any um, lipid, your cell membrane will solidify because they are phospholipids. You do not want it to solidify. So what cholesterol does is it creates areas that allow the cell membrane to stay flexible even in lower temperatures. Do ribosomes on the rough ER only make, yes, they only make proteins that are going to be secreted. And the reason for this is if it's going to be a protein that's going to be used on the inside of the cell, it's just going to be transported to a ribosome that's floating around on the cytoplasm. The endoplasmic reticulum is part of the endomembrane system. So it's basically the system of membranes on the inside of the cell. So that includes the smooth ER, rough ER, Golgi apparatus, and then the actual cell membrane itself. Because what's going to happen is those proteins are going to be created. They're going to go through the ribosome directly into basically a pod, a vesicle ready to go. That vesicle could get shipped to the Golgi just to finish kind of folding up that protein before it's sent out. But it's going to be put into its own little pod so it can leave. Right, until we wait for another thing. Disadvantages of cholesterol is as it even though it makes the cell membrane really fluid, it creates little like gaps. Which I mean, a few gaps are no big deal. You know, a few gaps are no big deal in the cell membrane. But if you have too much cholesterol in the cell membrane, although you'll have a really fluid cell membrane, there's also going to be areas where particles can get in and out without any supervision from that cell membrane. Saturated versus unsaturated. So saturated fats are like casasola. So saturated fats happen when you have, I don't have any paper examples. So actually, let me pull up a picture of it for you. Yeah, pictures are good. So all the images being pulled up. Saturated fats are completely saturated with hydrogen ions, or with hydrogen in general, where unsaturated are not completely saturated with hydrogen ions. So let me find an image. Remember, these are hydrocarbons. So it's just carbon and hydrogen. Open image in new tab. Here we go. Pop. All right. So this top one's a saturated fat, and so you can see every single carbon. It makes 4 bonds. It makes a carbon. It makes a bond to the carbon to the left, the carbon to the right, and then a hydrogen and a hydrogen. All right. An unsaturated fat will sometimes have double bonds in between the carbons. I think they can even go triple bonds. I don't think I've ever seen an example. I'd assume they can because, triple bonds. But what's going to happen is because of the way that these, man, talking's hard, these chemicals interact. And so think back to chemistry when you're going over shapes that, did you guys cover this? Or is this more like an OChem thing? It might be more like an organic chemistry thing. Anyways, so let's go back a little further step. So when you have molecules that have been bound together, they're trying to keep the maximum distance apart from each other. And this is due to the electrons on the outside of the elements. And so they are repelling against each other. So they're going to try to repel the maximum distance each. So if you have two, just 2 carbons next to each other, the farthest distance they can get is directly across from each other. But if we add in an extra element of any kind, let's say hydrogen, now there is going to be they're gonna have a different shape in which it creates based on they're trying to stay away from each other. Now, the hard part about this is imagining them in a 3D space inside your brain. And this is the part I always had trouble with is, okay, if they're a 3D environment in the brain, and so the problem is if you have your carbon, your 2, let's say carbon on the other side, it's going to be a straight line. But if you had extra an extra element attached to that, they're going to end up being, I believe it's 120 degrees difference from each other. So basically, they're going to kind of look like almost like a triforce. So we have one on the top, middle, and then two on the bottom. That's because that's the most they can space out. However, if you have four bonds, like in the case of saturated fats, it's going to end up kind of looking like a tripod. So you're going to basically have like three and then one sticking up. Now, the difference between having three like this, because you can see my fingers coming towards you, this thumb is coming away from you, versus like this, ow, my fingers don't stretch that far, will actually change the shape of the whole molecule itself. And that's exactly what's happening in your unsaturated molecule, is because those carbons are double bonding, now the physical shape, the way those elements will orient themselves in 3D space, think about that for a second, physically changes the shape of unsaturated, which is probably more information that you guys need to know. What you should know, just ignore the rest of everything I just said. Saturated fats are completely saturated by hydrogens, and unsaturated fats will sometimes have double bonds, creating kinks in the chain, those hydrocarbon chains. So you need to know. Now, saturated fats are solid at room temperature. These include things like lard and butter. Also, coconut oil. If you've ever seen coconut oil, it is actually saturated fat. And that's why you have to like scoop it out of the jar. You just can't like pour it out. Whereas unsaturated fats are things like canola oil, or vegetable oil that you buy in the bottles. Olive oil is also an unsaturated fat. Now, interestingly enough, there is a third category of fats called trans fats. Trans fats are when they take unsaturated fats and they turn them into saturated fats. That way they don't have to label them as saturated fats. They can just label them as trans fats. How dare they? Then there was a push several years ago to eliminate trans fats from food because it was very confusing for consumers and, not good for you because saturated fats. As far as diets go, you want to limit the amount of saturated fats and use more unsaturated fats. Better for you. Doesn't, clog arteries and stuff. Sorry I got into the organic chemistry. I got too excited about chemical structures. Alright, anything you guys have? This might be our shortest stream yet. Instead of being 2 hours, this one might just be 1 hour. What? This is really awkward if you guys aren't asking questions. Alright, it's looking like that's gonna be just about it. See anybody else asking questions? And there's like 6 other people on. It's kind of the time to do it. Meiosis 1 versus Meiosis 2. Alright. So the difference between Meiosis 1 and Meiosis 2 is whether you are separating homologous chromosomes or if you're separating sister chromatids. So a homologous chromosome, or homologous chromosomes, I should say, is when you have chromosomes that aren't necessarily exactly the same, but they do have the same genes in the same positions. For instance, I'm making this up, this isn't exact, on chromosome 20, we'll go with, position 38 is going to be where eye color is. And so it doesn't matter if the chromosome's from mom or dad, eye color is at that exact location. Now, they could be different alleles, like mom's going to give you a brown eye, Dad's giving you a blue eye. They could be different alleles, but they're in the same position. Now, this is important because if you don't have it in the same positioning, it's going to cause problems later on. And this is why some species, even though they look really closely related, can't actually mate. It's because those chromosomes don't line up when the cell's trying to, after fertilization, is trying to go through mitosis to build more cells. It physically can't because the information's all wrong and all in the wrong spots. Anyways, so during meiosis 1 is when those homologous chromosomes, I like to use my fingers for this, so homologous chromosomes split apart, and then during meiosis 2 is when those sister chromatids split apart. Now, when you split homologous chromosomes into two, this is basically mitosis, all right? And the thing that makes mitosis meiosis differently is when you form that tetrad, when those homologous chromosomes are next to us, and you do crossing over, that's one of the differences. And the second difference is it will split again, okay? That way gametes have only one set of chromosomes per cell. And what that's going to do is it's going to cause each gamete to be genetically different from each other. So that way it produces the most variety in offspring, which is an evolutionary advantage, but evolution is not going to be on the test. All right, can you go over tertiary structure of the protein? Yeah, so tertiary structure is the interactions between the R groups of the amino acid chain. Don't scare me. So yeah, so all those different R groups you guys have, you know, they can be polar, non-polar, they can be polar, non-polar, acidic, basic. Sometimes they'll have special interactions like cysteine has a sulfur on it which likes to produce disulfide bonds. And so if you have two cysteines really, really close to each other, they're likely to bond to each other. We call it a dicysteine bond. The cysteine's one of the amino acids. So tertiary structures when those R groups come into play. All right, I got a text from Lewis. Hey, Casa, so I'm a little behind on the live stream. You said you have all the papers and like equations ready to go. Where do I find that? So most of the stuff is actually already been posted to Google Classroom. The equations, let me head over to Google Class real quick. I believe were posted on our first live stream. Second period, YouTube live stream. Do's and don'ts, FRQs. If not, you can always just go to the College Board website. And they will have them. They have them on their site. You just put AP Bio in College Board, and then they will. Where is it? After Earth Day. Live stream time? Nope. Oh, man. The AP test info. Alright. Just, oops, too many tabs open. See, okay. So to get to the equation sheet, I just put in AP Bio equation sheet. I mean, I even spelled bio. I'm so lazy. AP Bio equation sheet. College Board. I don't know what that first link was. It was weird. But it'll pop up right here. I know I posted a link recently. I don't remember where I posted it, but I did post it. Alright. Pull this back. Close all these out. Too many open. So we have a question about the photosystems. Why are you so tiny? Ew, that's a terrible picture. Alright, let's go. Oh, I like this picture. No, I don't. That's a terrible picture. Oh, here's our picture from class. Bop. All right, so photosystems. So one thing to think about for photosystem is photosystem 2 comes before photosystem 1. Just so you guys know. All right, I'm actually gonna head over to my AP Bio book again, just to make sure to see exactly what you guys need to know about the photosystems. All right. Okay, cool. So, the essential knowledge of this particular question, which is photosystems, is going to be photosystems 1 and 2 are embedded in the internal membranes of the chloroplast. So you need to know what organelle they're in and know that they are a membrane protein. And they're located in chloroplast. And they transfer high energy electrons through an electron transport chain. Here is the electron transport chain, actually right here in the middle. And then there's a bunch of little other bits that you probably don't need to know for this, like the P680 and the P700. Those actually deal with the frequency of wavelight that they react to. Yeah, so it's like, I don't remember the units for wavelength of light, but yeah. And so you have all these different like chlorophylls, alpha chlorophyll, beta chlorophyll, and light comes in, excites those electrons. Those electrons basically make their way through to the primary acceptor, which will then go through the electron transport chain, back to photosystems 1, to primary acceptor, and then it will it'll get one of those NADP pluses to turn into NADPH, which that carrier will then go on to the next step of photosynthesis, because this is part of the light reactions. So that NADPH will then head over to Calvin cycle. Over to Calvin cycle. Ooh, I can't type for beans. Oops. This one. Here. Here. All right. Max asked about the tertiary. Yeah, no, tertiary is the interactions of the side chains. Tertiary is interaction of the side chains, and that will end up forming a globular unit of some kind. Because remember, secondary is the hydrogen bonding that happens, and that forms the beta pleated sheets. They look like pleats on like a skirt, like a tennis style skirt, and then the alpha helices. So I think you were thinking about secondary, but it's actually tertiary is it's forming a globular unit based on the interactions between the R groups. Yeah, photosystems are in the light reaction. And one of the things to keep in mind with the light reaction is, well, it only happens when there's light. But this happens in the membranes of those thylakoids. So it's not happening in the membrane of the chloroplast itself. It's happening in the membranes of those thylakoids. And remember, photosystem means light. It's a light system. Here we go. Open image and that's alright. So this one's for you. So yeah, so primary is just the chain, secondary is the alpha helices and the beta pleated sheets, and then tertiary is the interaction between the R groups to form a globular unit, And then quaternary is when you have more than one globular unit coming together to make that final protein. There's actually a lot of proteins that are made up of more than one part. For instance, although not made out of amino acids, made out of rRNA, ribosomes are actually multi-globular units. But it is RNA and not amino acids, so. Anything else for anybody that's popped in? The chat's open on down below. That way, I think. Down about there. All right. Any other questions? Oh, some other things to do before the AP exam starts. Aside from getting your equation sheet, getting out all your paperwork that you're needing, making sure you have a nice place to be, a quiet place, maybe hook directly up in the internet, Wi-Fi, whatever, where there's a strong Wi-Fi signal. Making your family aware that, hey, I'm taking a really important test. Can you please just shh? Sometime Sunday, take a break. All right, you're not going to do yourself any favors by trying to overload your brain. So I don't know, watch episodes of The Office, maybe binge like 4 episodes in a row. Okay, do something for you. Maybe take 20 minutes and paint your nails, or take a bath. I don't know, read a book, although not a bio book. Okay, take some time to just relax. Because one of the problems is I don't want you guys to overload yourselves. And get too stressed. Like, one of the things I learned about myself is I can't study within an hour of a test. It just freaks me out, and I just get really, really nervous, and it just doesn't work for me. So I can study. I taught myself I have to study up until an hour before any exam, and then I have to put it away. I can browse the internet for funny cat videos, or I can, watch an episode of The Hot Ones or whatever, but I absolutely cannot study in the hour before a test. It just makes me crazy brained. Okay. I'm pretty sure. Okay. I'll answer that later. Alright. Let me get into this. Pull out the. We're looking at SNRPs and RNA. Don't think those would be on the test. Those are pretty extraordinarily specific. Those actually don't look like they will be on the test. I don't see them anywhere. But again, those are also very, very specific questions. Let's try a different unit. Nope, I don't want to be in this unit. Alright, so SNRPs, it's actually I pronounce them as SNRPs, they help to form spliceosomes, which are responsible for cutting out those exons and introns. or cutting them up and then allowing all your exons to be put together. Remember, introns are non-coding parts of the DNA. So that's what SNRPs are. Although I would assume they'd probably only ask maybe about spliceosomes, which turns your pre-mRNA into just mRNA. Ooh. Nope, I'm not reading a scientific paper right now. Yeah, don't even worry about an RNA. Bit above your pay grade, Gassan. Squeaky. You're welcome. Alright, so Monday is our task, the 18th. Good luck. I think you guys are going to do fine, honestly. I think maybe the format might trip a couple people up, but as far as information goes, you guys are a pretty bright year. Pretty darn smart. Nope, not answering that. RNAi. I don't think interfering RNA is going to be on the test. Oh, some sad news. It's looking like your guys' class is going to be the last class that got the summer work. Because, you know, it's kind of hard to give out summer work right now. Now, what's it? Nope. Make sure you eat a good breakfast on Monday. Although the test is out. Have a good breakfast. Get some protein. Keep you fuller longer. Some carbs. Well, not too many carbs. Alright. Heterochromatins. So chromatins deals with DNA. Hetero is different. Images. Just find a picture to pull up for you guys. Oops. Give me an image. Got it. There we go. Open image. That's alright. here is an image of heterochromatin. And so heterochromatin DNA is not active DNA. The reason for this is it is very tightly packed. This is very common in interphase, typically during at the beginning. And it also has long stretches of repeated sequences. If you look at the ends, telomeres, these are, I think they're A's and T's, adenines and thymines, long repeating ends. Basically, they're caps for the DNA to protect it. So as DNA is being replicated, it always falls short at the ends. It makes it just a little bit shorter. So you can actually pretty accurately gauge the age of somebody using only their DNA, because you look at the length of telomeres. Now, unfortunately, as you grow older, those telomeres shorten and could actually end up eating into the actual coding part of the DNA, which could cause some problems later on. So heterochromatin, look at the orange regions. These are not active, and it's also great because these are the areas that get beat up the most. I mean, around the centromere. So when those spindle fibers attach, to cause a little bit of damage, who cares? And then also, again, telomeres, which are designed to protect you. There's actually an enzyme called telomerase that lengthens these, and there's actually a couple organisms in the animal kingdom that actually are allowed to turn telomerase back on, which is something we can't do, which is quite fascinating. And they can actually unage themselves. There's a little polyp that does this, and it goes between its two parts of its life cycle, between like baby and adult. And then when the adult gets too old, it goes back to the baby, and then to the adult, and then to the baby. We're actually studying that to figure out, like, hey, can we do that too? Do like some serious Benjamin Button. All right. Oh, and then the euchromatin. U means true. This is generally more active, and in interphase, it's generally not condensed, that part, because it has a lot of stuff that we need to access at any given moment in time. All right, any other questions? You guys don't have to wait for me to be done with one question to post another question. I'll just get to it eventually. All right, it's kind of looking like that's going to be the end of it. Guys, get any more questions, please feel free to email me, put up a thing on Google Classroom, send me a reminder, so long as it's within normal business hours. Do not be texting me at midnight. You know who you are. All right. Also, please let me sleep until at least, 6:37. I'd appreciate it. Let me sleep until 7:25. The exact time. 7:25. All right. Well, that looks like it's gonna do it for this episode of Hermitcraft. I mean, Casa Solo Classroom. Too much internet. Alrighty. Again, let me know if you guys have any questions. Good luck on Monday. I believe in you. You guys can do it. Don't worry about it. Don't stress. It's just one test. And honestly, considering the circumstances, a lot of colleges are going to be pretty understanding if you don't pass a test. And honestly, I didn't pass my AP test, and I'm kind of glad I didn't, because I got to enjoy a lot more of the higher level college AP or college like bio classes, and like some of the labs we did in the opening bio classes were things we didn't get to do. and things we still don't get to do at the high school level. So that was kind of fun. I mean, there's always a silver lining, and I thoroughly enjoyed all the professors I had, and yeah, so don't worry about it. It's all good. And if you fail, you're gonna end up being an AP Bio teacher. Think about that. You're gonna love your job. You're gonna go to work every day with a smile on your face. And what a horrible fate that is. What a horrible fate that is. All right. Best of luck to you guys. Again, if you need anything, let me know. All right. We'll have to definitely set up a time next year to cook AP Bio pancakes since you guys didn't get it this year. All right. Have a fantastic rest of your night. Good luck on Monday. I believe in you guys. All right. Bye. And you're welcome.